Welcome, everyone, to our grand rounds. I guess a lot of people have started their 4th of July weekend early, but we're very lucky to have Dr. Bernier, is that how I'm pronouncing it?
That sounds perfect.
All right, as our speaker. And here we have his introducer.
I'm very honored to be the introducer. I'm Mendy Minharas. I am a clinical psychologist over at the Autism Center, and I work regularly with Rafe, so I have the pleasure of introducing him today. I've actually prepared a few remarks. I don't always do that in situations like this. Rafe has a long and illustrious list of accomplishments, and I wanted to make sure to do it some justice. So I'll start out by maybe just giving you guys a little flavor of who Rafe is, if you don't already know him. He has a little bit of a reputation at the Autism Center. You will definitely understand a few of these references as soon as you hear him start speaking. But over at the Autism Center, he often gets called a hummingbird. And that is because he is always flitting around doing so many things at once that nobody can keep track of him. We have a few ground rules for when he speaks, so I'm going to rely on you guys to help us enforce those. One of them is to please steer clear because we don't want you caught in the crossfire of his gestures. We also want to make sure that he keeps both feet on the floor. That's a rule for his safety as well as yours. He gets quite involved in his speaking, but I think that's a reflection of the passion that he has for his work. When I asked Rafe about what a few key highlights might be for kind of an introduction for him, he sent me a very modest list of accomplishments that I knew didn't actually capture many of the impressive things that he's done in his career. And then he concluded his email by saying, or you could just say none of that. So I'm going to highlight a few of those things and add a few more that I think do justice to the kinds of accomplishments that Rafe has had. He did his undergraduate work at Tufts. He received his PhD from University of Washington and completed his internship at UCLA. He's also spent time in the Peace Corps. And he wears many, many different hats. He's a clinician at the Autism Center. He's a scientist with an impressive sort of history of NIH funding, and a professor who teaches regularly at UW and mentors quite a few students. He's also the interim director for the Seattle Children's Autism Center. And recently, one of his most recognized accomplishments is he's the PI on a national multi-site study, the Safari Spark study, which is going to be the largest banking of DNA from individuals with autism that's ever been collected. And you guys may have heard about that in the media. It's gotten quite a bit of press attention recently. But that's a really, really exciting project that's going on right now at the Autism Center. Anybody who attended the recent psychiatry faculty meeting knows that Rafe also recently received an award for being an outstanding mentor for the Department of Psychiatry. So I thought I might read a couple of quotes from people in his lab that give you an idea of what kind of a person Rafe is. So one person said, Rafe currently serves as a mentor for 31 individuals in our lab. And that's just his lab alone, let alone the rest of the Autism Center. That includes 9 undergraduate research volunteers, 7 full-time research assistants and coordinators, 7 graduate students, 3 postdoctoral fellows, a senior clinical psychologist, and a junior faculty member. And in querying the lab to participate in the nomination, the response was immediate and unanimously positive. This is exemplary of his unique and important role in all of our professional development. Someone else said, it's extremely rare to find a person who's mastered the tricky art form of balancing an ambitious vision for scientific productivity while remaining steadfast in creating time and opportunities for others to ensure the success of those around him. Rafe is this type of leader. Despite the immense demands of overseeing a productive lab at the UW and being the clinical director of the Seattle Children's Autism Center, Rafe's genuine desire to make time to support us as we pursue our own interests and our own skills is uncompromising. Rafe's door is always open. He never shies away from a chance to meet over a challenging data set or over a beer to discuss professional development and life decisions. I think that speaks volumes from Rafe's team about what kind of a person he is to work with. So without further ado, I'll turn the floor over to Rafe. Please keep your feet on that floor, if you will. And the title of his talk is A Genetics First Approach to Parsing the Heterogeneity of Autism. Thank you.
Wow. Thank you, Mindy. Maybe we could just stop right there and we can all go on vacation, because I'd like to end with that. That was a wonderful way to. Thank you, Mindy. That's very kind. All right. Great. Well, thank you guys. I'm very happy to be here today. This Friday. Excited to talk about kind of my approach to thinking about how we can parse the heterogeneity of autism. Before we get started, I just want to quickly highlight no conflicts of interest, nothing to disclose. My funding comes from NIH, Simons Foundation, Autism Speaks. Get a few pennies from the sale of my book, and that definitely would not count. The amount of that would not count as a conflict in any capacity from anyone's perspective. So what I want to do today is kind of start by talking about the behavioral definition of autism and how we've tried to parse that heterogeneity behaviorally, and then really move to discussing my journey through understanding the genetics of autism and thinking about parsing that heterogeneity from a biological perspective. And then I hope to kind of pull that together at the end with some clear take-home messages about how thinking about parsing that heterogeneity from a biological approach has more translational utility to aid in clinical care. And I hope we can sort of walk away with that message at the end of this talk. So there's a common phrase in the autism world: you've met one child with autism, you've met one child with autism, just speaking to that, the broad heterogeneity we see in any given patient. A good example would be just not too long ago, just a few weeks ago, I had two kiddos that came in back to back in the clinic to see me. First kiddo was a youngster who was minimally verbal, had a comorbid diagnosis of intellectual disability, developmental delay, and had significant seizure challenges, complicated medical challenges. The very next hour, another kiddo, also with a diagnosis of autism spectrum disorder, came through, was a highly verbal youngster, had an IQ well above the average range, and sort of mom was saying, this kid never gets sick. Wow, we've never been to the doctor because this kid's so healthy. And so it just speaks to me, in that 2-hour period, I saw two radically different children, both with diagnosis of autism. And that's not lost on folks in terms of, we've been thinking about this broad spectrum for a long time. We've come up with some good, clear behavioral definitions. You know, both of these kids met the clinical criteria in the social communication and interaction domain, and they also had the presence of symptoms in that second domain, the restricted and repetitive interests and behaviors domain. We need to come with a better name for that domain. At any rate, both kids were similar in that realm, but still had great heterogeneity, and folks have known this for a long time. Back in the 80s, Wing and Gould had come up with an approach to try to subgroup these kids behaviorally. They came up with 3 terms. You could group kids with autism into a group of kids who were aloof, a group of kids who were withdrawn, or a group of kids who were active but odd. And that started to get some attention in the literature. And then American Psychiatric Association started putting together Dsm-4 and said, hey guys, those are pretty cool terms. They're kind of nice, they're kind of catchy, but we're going to go with different behavioral subtypes. We're going to sort of subgroup this autism spectrum to autistic disorder, Asperger's syndrome, and PDD-NOS. And for roughly 2 decades, there was a lot of work done thinking about these 3 behaviorally defined subtypes in autism. And in those 2 decades, we found a couple of things. First of all, that there was no clear etiological indicator that's sort of differentiated by these biological subtypes. Nothing that was sort of jumped out there. There's also no good clinical utility from these biological subtypes. That is, if a kid came in to see me and went with a diagnosis of autistic disorder, I wouldn't say, oh, you're going to need this specific treatment. But you, Joey, because you've got a PDD-NOS diagnosis, sorry, totally different treatment. It's going to be specific to that. So there's no clinical utility in that realm based on these behaviorally defined subtypes. And nor can these even be reliably defined and utilized in a clinical setting. We'll talk about that in a second. And so that's why really there was this a move in 2013 with autism to, in the DSM-5, to just say, you know, we're going to go with autism spectrum disorder. We're going to do away with these behaviorally defined subgroups. So one of the things that drove some of this was the work that we'd done here at UW as part of the Simon Simplex collection. Now, what the SSC was really a large-scale collaborative studies that were designed to focus on gene discovery. That was the focus of the study. It was supported and funded privately by the Simons Foundation. Jim Simons is a hedge fund owner who said, you know, we got to do something about this autism thing. Let's get people working together because you guys are doing great work up at UW, you're doing great work at Yale, great work at UCLA, but you guys got to work together to actually make any headway. So pulled together 12 academic institutions around the country to really focus on gene discovery. But what we needed to do in order to do this type of project is we need to make sure we're doing everything exactly the same way. We needed to be working together, truly. So we made sure that we're going to collect all of our measures in the exact same approach using sort of strict reliability criteria. So what we did is we would videotape everything we did, and we would make sure that what we were doing at UW was exactly what they were doing in Missouri, was exactly what they were doing up in New England, exactly what they're doing in Texas, to make sure that we were really collecting it in the exact same way. We also had very strict guidelines and criteria for how we were including kiddos in this large study of individuals with autism. And so what we did is we used two primary diagnostic tools, the gold standard tools for autism diagnosis. One is the Autism Diagnostic Interview, the other is the Autism Diagnostic Observation Schedule. And we had very clear rules on who was allowed to administer these measures and to make sure we were administering them exactly the same way across all the sites. As I said, we videotaped everything, and I can tell you that in the 5 years that we were doing data collection, I double-coded more ADOSs and ADIs than I ever want to for the rest of my whole entire life, because I was watching everyone at Harvard and all these people at UCLA, and we were all doing this across all the studies, but it really allowed us to be very clear and confident that, okay, we're doing things exactly the same way. We also did, the third criteria that we used was we had to make sure that, so folks had to meet criteria in the ADI, on the ADOS, as well as meet Dsm-4 criteria. Of course, it's a manualized approach, so we were all experts. We would use that manual to help to aid in this in terms of our diagnostic subtyping. So we collected a lot of different measures, hundreds of different measures, and we made sure we were doing things exactly the same way, and we were seeing the same types of kids. And sure enough, we were. What we have here on this slide are just examples from 6 of the measures we collected, again, of hundreds. And on the x-axis, we have the different sites. On the y-axis is that mean performance for a given site on a given measure. So upper left here, we've got our verbal IQ. And what you can see as you look across the sites is, the gray bars there indicate kind of the range performance. And so you can see we're all seeing the same types of kids. There was no one site, this is true for all the hundreds of variables we look at, there was no one site that was seeing kids that were, mostly minimally verbal. No one site had kids who were particularly irritable or had challenges with adaptive skills. We're all seeing the same types of kids. But then when we looked at how things played out in terms of diagnostic subtyping, things looked radically different. So this is based on the first 2,000 individual families that we enrolled, first 2,000 children with autism. And so you can see here, x-axis here is, again, sites. Site G, 100% of all the kids that they diagnosed, they decided met diagnostic criteria for autistic disorder as the subtype. Site L there was about 50%, a little less than 50% of kids met criteria for autistic disorder, about 40% for PDD-NOS, and a little chunk there with Asperger's syndrome. So it's radically different across sites. And I have to tell you, as a clinician involved in this project, I thought, wow, that's a bit of a drag. We're not really doing so well with this. When we look carefully at what's driving that, the factors were wholly idiosyncratic to that site. So we could look, because we had so many data points, we could actually see, oh, for example, site E, their decision point was really about how irritable kids were. And so kids who were particularly irritable ended up with a PDD-NOS diagnosis versus an autistic disorder diagnosis. Pretty idiosyncratic across sites. So our expert clinicians, in theory, these are supposed to be the top clinicians in the country. We can't seem to agree as to how we should subgroup kiddos. So for me, this was kind of the final message to say, maybe we really need to be thinking about a biological approach or a different approach to parsing that heterogeneity in autism. So I first got into autism back in the 90s, and I was involved on a project as a research diagnostician working for Susan Folstein. She had published the first twin study of autism back in the 70s, and then she did a lot of things like the mini mental status exam and was involved in Huntington's and a number of other challenges. But in that role as a research diagnostician, what I did was I was driving all over New England. I was using those gold standard tools, the ADI, ADOS, making diagnosis for determining inclusion for a multiplex study. That is a study where there were we included families if there were 2 or more children diagnosed with autism. And we're applying sort of the cutting edge genetic technology of the day, which is a linkage analysis, to see if we could identify regions of interest in the genome that might be harboring gene or genes associated with autism. So what I was doing, I drive around all over New England, do my diagnostic evaluation, whip out my phlebotomy kit, collect some blood, we'd look at the DNA. And I'm not going to spend a bunch of time talking about that early work, because we were doing it and a number of other folks were doing it and never really managed to get anywhere. That's the take home of that. So I'll spare you all the details, but the linkage study approach wasn't really helping out, nor was the genome-wide association approach really leading to some answers that we were doing. But anyway, so I'm collecting this data, and then Susan says to me, she says, "Rafe, this is great. You're going all over New England. I love it. We're collecting all these samples. But you know, multiplex families are really a minority of all the families that are impacted by autism. The majority of families are simplex families, families where there's only one child that's been diagnosed with autism. So I want you to get back in your beat up old car, and I want you to drive over New England again. I want you to work with families where there's one kiddo that's been diagnosed with autism, and then there's a sibling who doesn't have autism." So I said, fine. So I get back in the car, I'll collect all this data. And then I get married. And then my wife says, "Hey, we're going to join the Peace Corps." So I say, "Okay," like any good husband, I say, "Great, let's do it." So I trot off the South Pacific with my wife. And I'm just going to share with you a little bit of sort of, so you can understand some of my naivete. But I'm sitting there on our little leaf and stick hut in the South Pacific, and I'm thinking, holy cow, they're going to find the genes for autism without me. I've made a horrible decision. This is a ridiculous choice I've made. Anyway, go, 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 go. I sort of laugh when I look back now. At any rate, so while I'm in the Peace Corps, there's this rogue geneticist out at Cold Spring Harbor. And I call him a rogue geneticist because he's really bright. And really forward thinking, but a very interesting presenter. This is how he presents. And so what we found here with our studies are this and that sort of thing. And it's sort of crazy. It's hard to follow him as he's talking to the wall and not talking to you. And he's talking about 1,000 miles an hour, which I know some of us do that as well. At any rate, this rogue geneticist says, you know what, there's some new technology. We've been focusing on these multiplex families for so many years. Linkage analysis isn't getting us anywhere. And he calls up Susan and says, "Susan, hey, you and Rafe collected all these simplex families. Could I utilize that data? Because I'd really like to look at something new that we're thinking about in autism. We're thinking about copy number variation. So structural changes, microdeletions, microduplications that arise anew. That is, mom and dad aren't harboring these. They're arising anew or de novo. And there might be something to this." So using that technology, looking for copy number variations, what they found is they looked first at the multiplex families that we've been studying and other folks have been studying for years. And they found that about 3% of these multiplex families have these de novo mutations, compared to about 1%. Typical population has these de novo mutations of the size that they were originally looking at. When they looked at our simplex sample that I'd collected all over New England, it turns out the rate was much higher. 10% of that sample had these de novo mutations that were arising. And it really created a change when we think about what might be going on with autism. So that is actually what drove the start of the Simon Simplex collection as I was mentioning earlier. This rogue geneticist, his good buddy was Jim Simons, and he said, "Jim, what we need 2,000 families. Oh, scratch that, we need 3,000 families." We started with 2,000, advanced to 3,000 because we were so successful. And we need to use this to identify these de novo events. See a higher rate of de novo events. Let's use this to aid in our gene discovery. So we started the Simon Simplex collection, and what some of us were working on this. What we found when we did our first analysis was that there was one particular copy number variant that was shooting to the top. That was CNV at 16p11.2. When you include both the deletions and duplications at this region on chromosome 16. That counted for about a little over 1% of the sample that we were studying. So with 1%, that was the most common single event that we'd found for autism so far. So we thought, great, wow, super common. Let's actually study this. So I worked with the Simons Foundation. We developed another program out of this called the Simons VIP Project. And what we did there was we said, we're going to really find some good answers for what's going on with autism by studying everyone that has a 16p11.2 event. This is so great. We're really going to find some answers. Again, here's some of my naive optimism that I'm sharing with you guys so you can see. So we set up a really beautiful online portal so that families could connect with us. We spoke to a gajillion medical genetics clinics and medical geneticists around the country and said, we're interested in you directing families to us if they've been diagnosed with 16p1.2 CNV. And then what we did was we invited families out to spend 2 or 3 days with us here in Seattle or 2 of our partner sites in New England and in Texas. And we were going to collect a lot of information about these families. And so I won't go through all the details, but we had a huge battery that again lasted about 2.5, 3 days, depending on the family, when we collected information from the child with 16p1.2, as well as any other siblings, whether they had the event or not, as well as any parents, or whether they had the event or not, including imaging, neurology, dysmorphology. We collected biospecimens. We collected a huge neuropsych battery. Well, it was a kind of a monumental effort. And what was interesting for me, as I sat down with the first kid that rolled into the study, he was a kiddo that had been referred to us from Texas. He was a 15-year-old young man. He had a diagnosis of autism. And as he walks to the door, I'm sitting there, I'm doing my evaluation with him. I'm thinking, you know, this really doesn't, this is not speaking, this is not autism to me. And so I really started pursuing further and trying to get a sense. What he was really presenting with was kind of a low average IQ. He had a speech sound disorder challenge. He had some language challenges and some motor difficulties. And it was just interesting. I could see how there would be a diagnosis for autism, give some of this presentation, but just didn't quite have the essence of autism there. In Go, we went on to proceed to see over 200 individuals with 16p1.2, over 100 deletion cases and over 100 duplication cases. And we saw that's a similar pattern. We're not seeing autism at every corner, which is what really we're kind of hoping to do. Instead, we found was a different presentation. So I'm going to highlight here just a quick summary of the roughly 200 individuals we saw. So it turned out that just under a quarter of our deletion cases and about a fifth of our duplication cases ended up with an autism diagnosis, and that is using clear ADI, ADOS, clear diagnostic criteria. But instead what we found in terms of our most frequent diagnoses, first for the deletion cases, was this pattern, this combination of a speech sound disorder, developmental coordination disorder, DCD, and a language disorder, their mixed expressive receptive language disorder. And that was really kind of the pattern for most of our kids. Over half of our kids ended up with this articulation challenge, speech sound disorder. Duplication case had a different presentation, with intellectual disability being the primary, most common single diagnosis, but we still see that pattern of developmental coordination disorder and attentional challenges. Many of those kids across whether deletion or duplication had a number of challenges. Almost all of our deletion cases had at least one psychiatric diagnosis. But there were individuals we saw who did not meet criteria for any diagnosis as well. So really kind of thinking about heterogeneity, even within this one particular subtype, we had a great deal of heterogeneity. The couple of things I'll highlight before jumping ahead is that there are a couple of interesting mirror phenotypes we found when we did this in-depth analysis of all these many individuals, children and adults with 16p1.2. Our deletions, we had a very high rate of macrocephaly, where we saw a higher rate of microcephaly in our duplication cases, higher rate of obesity in our deletion cases. In our duplication cases, we actually found several cases with failure to thrive and many underweight kiddos. Relatively high rate of seizures in both groups, but almost a third of our deletion cases ended up with a seizure history. Interestingly, this is more observationally, but when I was spending all this time with these kids, I realized even the kids that didn't meet criteria for autism still had significant social challenges. So one of the things we looked at is we looked at standardized quantitative assessment of social ability in the parents and the siblings and the kids. And one of the things we found when we quantify what those differences are is we see a significant shift here. What we're seeing here on the x-axis is as our scores on the social responsiveness scale. It's a questionnaire that looks at social relatedness, social ability, social motivation, social cognition, and other sort of autism-related symptoms. And it's appropriate and normed for adults and children, so we can actually use it across our sample. And what we see here is we see almost a 2 standard deviation shift in terms of social ability, regardless of the diagnosis of autism in our individuals 16p1.2. We're using our siblings and the siblings and parents as essentially our control to think about what we'd expect. We'd expect, based on our parents and siblings, that we'd have a pretty low SRS score. And we see that shift in red for our probands, our individuals with the 16p1.2 CNV. We see that shift to a higher score, indicating greater social impairment. So I highlight this just to say that even though we weren't necessarily seeing autism in terms of a diagnostic threshold, at a quantitative level, we're still seeing a lot of social challenges in these kids with 16p1.2, kind of across the board. Yes, oh yes, question.
Just if we looked at just siblings or parents and their children,
Mhm.
like the green and the blue, and just unaffected families, those two curves are very different. I'm just wondering if there's something unique about what goes on in children.
In the sibling. Okay, great question. Right. So just the question for folks that might, somewhere out there on the Ethernet. So the question is, we've got, looks like different curves for our parents, for our siblings, for programs really have kind of a differences. There's something specific about kids that looks a little different for the siblings, why you have that tight curve. And that's a good question. I don't actually know a good way to answer it. There's the measures and metrics we were using are normed metrics. So when we think about children generally, we wouldn't necessarily think about such a tight curve in siblings. What my guess is truly what's driving that is that parents are filling this out both for their children who have CNV and their siblings who don't. And my guess is of some sort of, a comparison effect there that's going on. So it drives kind of they're really not quite as impaired. That's my guess. Yeah. Oh, the question in the back as well. Yeah. So the deletion is quite a big chunk. Now, it's 29 genes localized there to this particular region on 16p1.2, 550 kilobase pairs. It's a relatively big chunk of DNA. Oh yeah, right. Yeah, perfect. Great. So this is kind of what we started with, looking at copy number variation. But then we're able to apply some new technology to think about this in a little different way. So as this is ongoing, as we're exploring copy number variation, we're able to get some pilot funds to put together our first exome sequencing study in autism. So here I work with Evan Eichler's group, and we started working together to think about how might we utilize families that we've collected to think about identifying some de novo mutations that aren't in specific in terms of impacting structure, but impacting particular genes to really kind of hone in the mechanism. So we identified 20 individuals with autism who were from simplex families and where there was no family history of autism anywhere, where parents didn't even have any sort of sub-threshold symptoms for the broader autism phenotype. We looked for families where there's really no mental health history at all, and where kids with autism who had been diagnosed were pretty impacted by their symptomatology. We identified these 20 kids and did exome sequencing on the kiddos and their parents. At the time, this was a relatively expensive $500,000 sort of pilot project back in 2009, 2010. But it was quite informative. So we selected our 20 individuals, and what we found in that group of 20 kids with autism that we tightly selected was we found 4 neurologically expressed genes that were disrupted. Where the gene was disrupted, it wasn't creating the protein it was supposed to create. These were mutations that were arising anew. When we looked at those 4 individuals, they seemed to be more impaired, both in terms of cognition and top, as well as in terms of what we call the calibrated severity score, or comparison score, which is a kind of a gross metric for autism severity. And I say gross kind of in many different terminologies. I mean, it's kind of gross, not very good. But at any rate, those folks were more impaired. But what this did was it provided us with the opportunity to actually convince folks that we could actually do this as a meaningful approach. So we redid the same exact study, but with 209 individuals with autism. And here, there was a lot of enthusiasm. We're gonna nail it. We're gonna figure it out. We got 200 kids. We've got exome sequencing on them and their parents. This is gonna be amazing. And it was cool, but didn't answer everything for us. So what we found was in our sample of 200 folks, about 25% of those kids had 1 or more de novo gene mutations that were neurologically expressed, that were disrupting the function of that development of the protein. What was a bit of a drag, I have to say, was that there were only 2 genes for which there was any recurrence, CHG8 and NTNG1. And so we thought, wow, that's really not as informative as we hoped. And so thinking about the number of potential genes that could be disrupted given our sample size, our estimate was there somewhere between 400 and 800 different genes that are likely playing a role in conferring autism risk. We published this paper in Nature. The same issue, there was another group that did a very similar type of approach, and their estimate was close to 1,000 different genes that could be disrupted that could be playing a role in autism. So that was a little bit of a but, sort of eternally optimistic. And what we did is we looked at some of these genes that were disrupted. And it turns out that many of them were all part of an interactive protein-protein network. And so we thought, we then sort of approached Jay Shanduri and said, hey, Jay, you've got this new technology using MIP sequencing. Perhaps if we targeted these genes that have come together in this network in a very large sample, because if we can target the genes as opposed to doing full exome sequencing, we can do this much cheaply, much cheaper. Let's try that. And that's exactly what we did the following year. Was used sort of a molecular inversion probe sequencing to target those genes that were part of this network in a very large sample, over 2,000 kids with autism, because we could get it down to about $100 per gene per kid, or per individual, I should say. So it allowed us to sequence a large number of genes in a large population at a fraction of the cost doing it with exome sequencing. And here we finally found some recurrence. We were very excited. We found an incredibly common gene mutation. There were 9 kids in our sample of over 2,000 kids that had the same gene mutation. We were fired up. Holy cow. Those were 9 kids with a mutation of CHG8. We also identified a number of other recurrent genes, DYRK1A, PTEN, SCN2A, and several different genes that have since become sort of high confidence genes. But what was interesting for me was that when we looked at these particular kiddos with these shared gene mutations, we started to see some patterns in the phenotype. Because we had such a large sample, one of the things we could look at was head circumference. It was a relatively easy metric that we had, phenotype that we had. So what we have here in this figure is z-scores, head circumference z-scores on the x-axis. In the dotted line would be kind of the normal distribution. Our shaded gray area here under the curve is what our total sample of just over 2,000 kids look like, a slight shift, which is what we see in autism broadly, so it mirrors that. And then in blue here, we have our 3 individuals who we identified with a DYRK1A mutation, all 3 of them with much smaller heads, macrocephaly. In red are our kiddos with the CHG8 mutation who had much larger heads, several with macrocephaly. In black is our kiddos with PTEN who had very large heads. So from my perspective, we're very excited about this project because we had, first we had some recurrence finally, and we could identify some genes we think were playing could be more confident about the role. And secondly, it was tied to the phenotype. So what was interesting particularly about these kiddos with CHG8 was that 2 of them happened to be from here in Washington State that had come into the lab. And so I called them up and said, hey, we found something kind of interesting. We'd love to have you come on in to the lab. We think that we've identified a gene that we think is playing a contributory role to your children's autism. Would you come in? They said, of course, we're happy to come in. Boom, they came in just next week. I was able to see them back to back over the period of a couple of days. And the first girl that walked in, a 12-year-old girl, cute as a button. So she walking into the lobby. She clearly had autism, kind of do what we call the LADOS or the lobby ADOS, where you just sort of realize, yep, okay, that kiddo clearly has autism. And as we sat down and did some testing with her, she had intellectual disability. As I'm sitting there, she had macrocephaly, and as I look at her, she had some slight hypertelorism. She had a prominent forehead, downward slanted palpebral fissures, subtle, but sort of make a note of that. As in chatting with mom and dad, she had significant sleep challenges. Sometimes she wouldn't sleep for 2 or 3 days at a time. Mom and dad would lock her into a tent, kind of lock her up so they get some sleep at night. I want to clarify, it was a very nice tent. They were very loving, but they just needed to get some sleep. Some sleep, and then had significant GI problems, gastrointestinal problems. These are 2 things we hear about a lot in the clinic with autism. So kind of made note, but I wasn't really going to write home about it. So I make note and go. The next few days, another kiddo comes in. He's an 8-year-old boy. And same sort of thing into the lobby. Yep, he clearly has autism. He did not have intellectual disability, which I think is relevant. But also, again, we see the macrocephaly, a prominent forehead, slightly slanted down palpebral fissures, and the hypertelorism subtle. And then again, in chatting with mom and dad, significant sleep problems, won't sleep for 1 or 2 days at a time, and significant gastrointestinal problems. So again, 2 out of 2. Again, I'm not writing home about it, but I'm going to make my notes. So go. We were able to see a total of 15 individuals or interact with 15 individuals from really around the world in that first year as we were pursuing this. And we found very similar patterns with these disruptions to CHD8. So we started with 15 kiddos, and what I think I want to highlight a couple points with this big, huge, heavy text-laden slide. So in our first analysis, we worked with 15 individuals, and we identified these folks from a number of ASD and developmental disability cohorts. We never once found a disrupting mutation to CHD8 in any of the siblings that we'd tested and any of the controls. We then replicated this the following year, where we expanded out. We identified more individuals, 22 individuals, and we've actually now up to 26 individuals now. And again, have not once found evidence of a disruptive mutation to CHD8 in any siblings or controls. So we're starting to generate some real confidence about the role of CHD8. But what we found phenotypically across all these individuals, so summarizing real briefly, is that all but 2 of the folks that we've identified meet strict diagnostic criteria for autism. The 2 folks that we haven't seen, one is a woman who's in her 40s who's been hospitalized in Sicily for many, many years. She has a diagnosis of schizophrenia and intellectual disability. Haven't been able to evaluate her. She's never had an ASD evaluation. I'm not sure what's going on there. And the second is another boy who has the same pattern, ADHD, attentional challenge, intellectual disability, as well as a diagnosis of schizophrenia in the Netherlands. He's not had a formal ASD evaluation either. So somewhere, in my perspective, somewhere between 23 and 25 of these individuals have a diagnosis of autism. Only half of them have intellectual disability. As I mentioned, we have some facial features, really recurrent issues with gastrointestinal challenges and sleep challenges. So this is all very interesting, but we really want to understand, well, so how can we be super confident that this phenotype is tied to this CHD8 mutation? So what we did is we worked with a gentleman named Nico Katsanis down at Duke University. And he said, you know what? I can do this in zebrafish. I can knock down CH8 in zebrafish, and we can take a look at the phenotype in zebrafish. So lo and behold, what he did was able to knock down CH8 expression in zebrafish. And what he found, he looked at head sizes in these little zebrafish. And what he found, is kind of hard to see in this image here, but up in the top left, we have our control fish, our normal little zebrafish, and the 2 examples on the right are various morpholinos. And if you measure their head size, they have larger heads. I don't measure fish heads, but I'm trusting Nico. He does this well and sort of in a strict scientific approach. But these fish have larger heads. So we started to be confident about that aspect of the phenotype. But Nico said, you know, I can do more though. Let me tell you what I can do. I'm going to feed these little zebrafish morpholinos, I'm going to feed them little fluorescent pellets. And we're going to track and see how long it takes for these little fluorescent pellets to pass through their bodies. And well, sure enough, these poor little zebrafish don't poo. And so I'm glad I don't do this sort of work. But anyway, this is the work that his lab is doing. And so here we have recapitulation now of at least a couple aspects of the phenotype that we could look at. Sort of challenges with the gastrointestinal system, specifically around motility and sort of this macrocephaly. We haven't gotten a really good assay yet for social interaction in zebrafish. But once we nail that, we're going to do that with zebrafish as well. So providing, again, more support that what we're looking at in this CHD8 disruption is really tied to the phenotype. One other thing we did was we also looked at where and when CHD8 is expressed in the brain. We want to make sure that, again, it makes sense and fits with our hypothesis about its relationship to autism. So what we have here is we have a couple different examples of brain regions. On the left, we have our dorsolateral prefrontal cortex, and on our right is our inferior parietal cortex. The age and x-axis and normalized expression values for CHD8 in red and CHD7 in black. And what we can see is that CHD8 is expressed very early on in development, 9 weeks post-conception and 13 weeks post-conception, kind of in that range, in 2 different regions that have been implicated in the brain in autism, whereas we don't see that same degree of expression in these regions for CHD7. That's kind of our control counterpart. So pulling those 3 bits of information together allowed us to feel really confident that what we're seeing here is we're seeing a clearly defined subtype in autism. And this is just a pictorial expression of kind of how we think about these things. we're looking at a genetically heterogeneous disorder. If we do targeted sequencing of a particular gene, we then make sure we're doing patient recontact and follow-up. Something we did, in this case, zebrafish modeling, expression analysis, and we can define a distinct subtype. And so what we then did was we actually put together a web page for families and clinicians to connect with. And that's since been built out to a really family-driven Facebook page. And so what has been nice for these CHD8 families that we've been able to work with, they've established their own community. A real nice way they can learn from each other and figure out, well, what's worked, what hasn't worked, provide some support. And it's brought these families together in a different way than having a diagnosis of autism has. So it's had some immediate clinical utility in a sort of thinking about it from a different perspective. So this approach, this way of thinking about the genetic contributions to autism, we did this with a whole bunch of other different genes following from our CHD8. That was really kind of our proof of principle. So we focused on kids with ADNP, DIRC1A, POGZ. We chatted about 16p1.2 in an effort to think about are there these genetically or biologically defined subtypes? And across all of these different particular genes, we started to find particular patterns. With our DIRC1A individuals, all of those individuals, all of them had intellectual disability. About half of our individuals had ASD, and some particular patterns we found in terms of facial features, issues and challenges with feet development, and feeding challenges only in development. We start to parse together a little syndromic presentation in terms of phenotype. So we've been doing that. We've been plugging along in exploring multiple different genes in the same sort of approach of targeted sequencing, phenotypic follow-up, and making sure in animal models and expression that's all coming together. And that's really been our model, and we continue to do that moving forward. And really where it puts us at is a very different genetic landscape from when I first started back in the 90s. Back in the 90s, what we would say when talking about the etiology of autism, we would say, well, we kind of know in terms of genetic contribution in about 4 to 5% of all kids with autism. These are things like Fragile X and Rett syndrome. We sort of kind of understand what's contributing tuberous sclerosis, neurofibromatosis. And we've really come a long way based on our CNV work, based on our exome analysis, and looking at doing targeted sequencing to look at specific gene disrupting mutations. We can now say that we can identify the genetic contributor about 45% of all boys and about 55% of all girls. And some of these are de novo mutations that we've been chatting about. We've done a lot of work with private events and recurrent events, as well as rare inherited mutations. And we've really made a lot of headway in terms of parsing these out. CHD8 being the most common single gene that accounts for about 0.3% of all kids with autism. 16p1.2, as I mentioned, is about 1% of all kids with autism. So we have this, so as we're identifying the particular genes that are involved, we can then start to think about, well, where are those expressed, how are they expressed? And we can start to think about then developing our targeted treatments. So it's just a little cartoon indicating some of the high confidence ASD genes that we identified, how they play out and in terms of neural structure and function. So CHD8, for example, is involved in chromatin remodeling. We know it as a gene that's turning on and off many other genes. That's its role. Interestingly, many of the genes that CHD8 turns on and off have already been implicated in autism and from other studies. So we've got a real convergence that CHD8 be kind of the master regulator of many of the autism-associated genes. Again, many of these genes, we won't go into detail with all of them, but some of them play a role in what's happening at postsynaptic density. SCN2A is an example of a sodium channel gene that's involved with neuronal function. With these identified roles of these proteins, we can start to develop targeted treatments. And that's exactly what's happening right now with many clinical trials are underway. I shouldn't say many. I would say few that are underway at utilizing particular compounds or existing medications with genetically defined subgroups as a way to start to think about precision medicine autism. We're still quite a ways away from actually realizing this, but we're starting out. So, but there are some more immediate clinical implications based on this work. The first is that in many ways for families, when we think about incorporating genetics work into the workup for an individual with autism, which is actually what the recommendation is from the American Academy of Pediatrics, that this provides some answers from families. And it really can provide some sort of closure on what's going on. more relevantly for us as clinicians, that it can help us to think about what we need to be monitoring in terms of ongoing care. So for example, with CHD8, we want to make sure we're thinking about GI symptomatology and getting good workup there, as well as monitoring sleep. I highlight the GI piece especially because we commented on this in our paper in Cell, but didn't make too fine a point on it. But we did work with 2 adults who had CHD8 gene-disrupting mutations. Both of them ended up having GI-related cancers, one of whom died from colon cancer. And so we didn't make too much of it, but those are the only couple of adults we'd seen, and here we're seeing this outcome. So again, don't want to jump to conclusions. We didn't make much of it in the paper, but the things we want to be monitoring for these individuals with CHD8. CHD8 has been implicated in some forms of cancer. As well with 16p, 1.2, again, we want to monitor for seizures. We've been able to develop some sort of practice parameters for many of our genes and copy number variants that we've been working on precisely for this. Think about what we need to be monitoring. As I mentioned, many of these families have been able to establish a nice community around the particular event. There's the Families SCN2A Foundation. I'm actually going out to speak with them in Chicago. This July is their first formal national meeting. Dirk-Winay, 16p12, CHD8, all of these organizations have erupted and developed where families are excited, enthusiastic, working together, sharing information, and in some cases driving the research. At a family mom of a kiddo with ADNP who lives in the state here, and she interestingly sort of had noticed that a bunch of her kiddo and a bunch of other kids had this really early tooth eruption. These kiddos were just a couple months old and teeth are popping out of their mouth. And she's like, what's going on here? Able to connect up with one of those folks on ADNP and developed a nice mouse model. And oddly enough, these mice, they've got early tooth eruption as well. So really interesting sort of, these are observations driven by parents that have been being replicated by scientists. So a real nice way of pulling communities together and driving science. And then really what's on the horizon is in guiding targeted treatments. As I mentioned, there's a couple medication trials that are already underway. There's medications that are already out there being utilized broadly, but if we target them, might have more utility. As well as one of the things we know about autism is that there's empirical support for behaviorally based treatments that's managing the core symptoms of autism. But we have no idea about, what we're not good at, is knowing who's going to respond to those treatments. So it might be that by understanding the biology better, we'll have a better understanding of who's going to be a treatment responder or not. So those are kind of the immediate clinical implications, again, on the horizon of these targeted precision medicine approaches. So just to kind of pull us all together here in the last couple minutes. So we have a couple minutes for questions. There's tremendous heterogeneity at the behavioral level, as I highlighted, and genetic heterogeneity as well. I'm talking about 1,000 potential different genetic contributors to autism. In this talk, we haven't even talked about some of the other risk factors that so we might consider environmental risk factors that might be contributing to this heterogeneity. So a great deal of heterogeneity in autism, but genetically defined subtypes, they show that there are very real but subtle subtypes there within this larger spectrum. And by identifying this biological mechanism, we can think about targeted treatments. That can help us inform and understand a course and prognosis, and for families provide a sense of community that can pull together. So this is just another pictorial representation of the way I think about this information and how to move forward with this. But most importantly, I think what really drives this is that working with families, and I always say thank you to the families, because these are families that give up a lot of their time to come hang out with me in my lab, sometimes for 2 or 3 days at a time, and hang out in Seattle, even in the dead of winter. Sort of thanks to my many collaborators, thanks to my lab, and thank you to you for listening to me sort of carry on here on a Friday afternoon right before a holiday weekend. So thank you. Yes, question in the back. Right. Okay. So the question was, are there individuals with CHD8 mutations that don't have autism, and what do they look like? So we have, so I'll sort of summarize this way. So we've not found anyone who is considered a typical control or a sibling at any database. We've not found anyone with a CHD8 mutation that doesn't have some significant impairment. Of the folks we've looked at and we've analyzed, 25 of those, we've looked closely at the phenotype for 25 of those individuals that we identified. 23 have had autism, have met strict diagnostic criteria for that. There have been 2 folks that have not been diagnosed with autism. I have not myself met these individuals. One is an adult with a diagnosis of schizophrenia and intellectual disability. The other is a young man, and she's been hospitalized since she was a child in Sicily. The other is a young man in the Netherlands, and he's also got a similar diagnosis, as well as an ADHD diagnosis. He's kind of got a smattering diagnosis, but again, intellectual disability and schizophrenia. Have not met this individual. Neither of those folks have had a formal ASD evaluation. So the answer I would say is somewhere between 23 and 25 out of 25 individuals with CHG8 have autism.
And those individuals, do they have certain phenotypes more than other patients with autism? I mean, in terms of the spectrum, something that makes them look different?
Right. So there are some features that make them look different, but not within the domains of autism. So one of the things we've looked at, so just to repeat the question, folks on the line. The question was if with the folks that do have CHG, do they somehow look different from other folks with autism? So there are associated facial features. There are the GI and sleep challenges that we sort of discussed that these folks have, a high proportion of folks have. And in addition to that, we've looked at within the autism symptom domains, social communication interaction and restrictive repetitive interests and behaviors. And what's tricky is we're not finding any differences on those domains for individuals with CHG relative to, say, kiddos with a DYRK1A mutation or ADNP mutation or WAC gene. Or I think really what that has to do is our level of analysis. We're really not so great at quantifying those challenges at a real fine-tooth, sort of a real fine resolution. Well, that's one hypothesis. The other hypothesis is that they don't really differ and it's all the same. But I think it's measurement error. The way we think about differences within those symptom domains, so we haven't seen anything specific. Except for SCN2A kids, those kids tend to be more socially impaired even on our gross levels of metrics relative to other kids with single gene mutations. Yes, question back there, yes.
Were the brain volumes increased in CHG8 individuals, was there a regional pattern to the brain increased brain volume?
Yeah, great question. Okay, so the question was any differences in brain volume with our folks with CHG8 and any sort of differences in that. So, so no gross differences in brain volume for folks with CHG8. Broadly, we didn't assess carefully. We didn't do any imaging in-house on that. We just looked at clinical MRIs and sort of got an assessment to take a look at, well, what's going on grossly. There's no gross abnormalities in terms of right. Didn't do any sort of analyses looking at sort of volume specifically, but nothing gross emerged for folks that looked at that.
And then the Allen brain now, the CHG8, you looked at 2 brain regions of interest.
Yes.
Probably expressed all over.
All over, true.
But that issue, I was wondering about the serotonin system, CHG8 expressed there.
That's
everything to do with the entire
nervous system. That's a great question. Yeah, so the question or observation is, might this have to do, the expression we should be thinking about serotonin systems. I totally agree. We haven't looked closely at that. We need to look more closely at that. That's a great point. Definitely. Thanks, John. Yeah. Yes.
Just to follow up on the midday question, to clarify that 23 out of 25 number,
yeah,
that's not based on a population sample in which you then identified who has the mutation and calculated it. Isn't there some collection?
Sure. So there definitely is going to be some measure of selection. So we've 100% totally agree. So the question was, is there some sort of selection going on with thinking about our numbers? Right. So if we start with our total numbers are that we've looked at thousands of kids, individuals sequenced as part of either research cohorts with autism or clinical cohorts with autism or intellectual disability. And we started broadly, sequenced all of these folks, and now even as of today, our sequencing numbers are even higher. But we've sequenced about 8,000 individuals, and we've identified in these, well, just under 8,000. In that, we've identified about 26 individuals with CHG8. So, it's not jumping out at us. It's still relatively rare. And we haven't found in any of our control cohorts that we've looked at, whether they're siblings, whether they're experimental controls. So we haven't looked at any sort of population prevalence of CHG8. But when we look at that big sample of almost 8,000 folks, and we look at that 25 that have been identified. That's where that 23 out of 25 number is coming from. So there's still some measure of selection because we haven't done anything at a population-wide level yet. Is that?
Out of 8,000.
Yeah, yeah. Great. Yeah.
So in your checking for mutations in CHD8,
Yeah.
have people looked at populations with macrocephaly or with GI problems that are severe?
Good question. Yeah. So the question was, has folks looked in samples of macrocephaly or GI issues? And that, to my knowledge, no one's done that yet, or no one's reported on that yet in literature. And I haven't chatted with anyone about that. Generally end up chatting with folks that are doing stuff with CHD8.
School of nursing has a lot of people they repeat other genes who have IBS. I mean, I don't know if
That'd
they
be
have
great.
numbers, but they have thousands of samples that they're
Here and yeah, that might be a good connection.
Cohen, was involved in that,
Okay, great.
down the hall.
Oh, good. Thanks. Yeah, all right. Yeah, I'll look into that.
Yeah, just talking about penetrance in general.
Yeah.
Has anyone estimated what proportion of autism cases actually have 100% penetrance versus ones that have been some sort of combinational or have a lot of environmental factors? Has anyone tried to estimate that number yet?
Yeah, so lots of folks have tried to think about this in different ways, and it's been really genetic event specific, I would say. There's some folks I've worked with have really tried to say, well, thinking about 16p specifically, thought, well, do we want to talk in terms of penetrance overall? Do we want to think about the impact? And that's kind of really where, when we were thinking about this concept, wherever I can get there, this concept of trying to quantify the impact of the event on a given phenotype. And this is the sort of notion, well, because how penetrant is 16p 1.2 in terms of autism? It's hard to say. Well, we can say, well, about in our large sample, about 23% of folks with deletion have it. But if we think about it in terms of a quantitative metric, we can say, well, gosh, in general, person per person, you have about a 1%, 1.7 standard deviation hit. If your folks are super social, you're not going to fall in sort of the ASD range because you're sort of, you're starting from a higher set point in terms of your social ability. But if your folks are a little bit off and you got the 1.7 standard deviation hit, you're going to be more impaired. So folks have started to think about it that way because it's been hard to look more broadly given how different and distinct each gene event is in autism. So I wish I had a good answer for you. That's kind of where it's at, state of the science. Awesome. Great. Well, thanks, you guys. 1 o'clock, we'll let you guys go.